Today we'll be covering genetic mapping. Here you can see an illustration of the chromosome. Throughout this video we'll be looking at the three related loci of the cat: fur length, color, and height. This cat can either have short or long fur, be a purple or blue color, and be a miniature or tall. It is important to always know what alleles are dominant and recessive. For our cat, the short fur is dominant over long, the purple coat is dominant over the blue, and the miniature height is dominant over tall. Our first test cross will be parents. We will use a homozygous dominant female and breed her with a homozygous recessive male. Next to each parent is our chromosomes. The first generation produced by the parents is referred to as F1. For our F1 generation, you get a cat that is heterozygous at all three loci. Below the offspring is a picture of its chromosomes. While the cat may be heterozygous, its phenotype is going to be that of the mother. The mother gave the offspring her dominant alleles, which match the recessive alleles inherited from the father. The F1 offspring will have short fur, a purple coat, and be of miniature height. Now we will take a female from the F1 generation and breed her with a homozygous recessive male in a test cross. By using a heterozygous female, we can ensure that the alleles given actually determine the phenotype of the offspring. The F2 generation may have offspring that take after the parents or offspring that experience recombination, meaning their genotype and phenotype differ from the parents. First, we will cover the single crossover between the S and P loci. This event produces short blue tall and long purple mini gametes. A single crossover can also occur between the P and M loci. This event produces short purple tall and long blue mini gametes. A double crossover event can also happen, and this event produces short blue mini and long purple tall gametes. In order to calculate the probability of each different phenotype produced in the F2 generation, we have to look at our genetic map. The distance is equal to the percentage of recombination between each loci. For our map, the distance between fur length and color is 17 centimorgans, and the distance between color and height is 6.2 centimorgans. The coefficient of coincidence we'll be using is 0.72. There are 8 different phenotypes that can be exhibited by the offspring. 2 of the phenotypes will resemble the parents due to no crossover event happening. These are called NCOs and are the parental types. These have the highest probability because it is more likely that the offspring will not undergo a crossover. 2 of the phenotypes will experience a single crossover event at region 1, and another 2 will experience a single crossover at region 2. The last 2 phenotypes will undergo a double crossover event. This event has the lowest probability because it is unlikely that an offspring will experience 2 crossovers. To better understand how I got these probabilities, I worked out a double crossover and a single crossover at region 1. To find the probability of the DCO event, you multiply the centimorgans from region 1 and region 2 by the coefficient of coincidence. Next, you multiply the DCO value by 1/2. You multiply by 1/2 because there's a 50% chance that the offspring could have exhibited the other DCO phenotype. You take this final number, multiply it by 100, and that is your probability of a specific gamete. Next, we have our single crossover event at region 1. To find the probability of this event, you take the centimorgan from region 1 and subtract the DCO value. You then take the SCO1 value and times it by 1/2. This product is then multiplied by 100, and then you have the probability for that specific gamete.